Okay, can see my slide?
Yeah, Professor, I can see your slides.
Okay. Assalamualaikum and good morning, ladies and gentlemen. First of all, I would like to thank to Universitas Brawijaya for inviting me as one of the speakers for the program on the initiations of the International Scientific Associations on Aquatic Environmental Health and Aquaculture. With the theme for today is enhancing aquatic animal health and ecosystem sustainability through One Health collaborations. As we know that aquaculture is important because it's among the fastest growing field in the food production industry and has become one of the main target for future food security. So today I'm very happy to share my knowledge, my experience from my research and also diagnostic work in aquaculture to ensure that fish or crustaceans such as shrimp, especially because I'm focused a lot on shrimp, are healthy and safe to consume. So, okay, before we go further into this topic, so my topic today is viral incidents in shrimp culture. Actually, this is a case study. I would like to introduce you to aquaculture. So, what is aquaculture? So, the definition of aquaculture in simple word is in simple sentence is aquaculture is mean that the farming of aquatic organism, including fish, mollusks, crustacean, and also aquatic plants. And why we are focusing on aquaculture is because the aquaculture is among the fastest growing field in the food production industry, as I mentioned before, and has become one of the main contributors to the country's economy. This is a very, very important. So, in any industry, so we have aim. Okay, what is the aim in aquaculture? So the goals of aquaculture for today and in the future is to provide society with enough food and that is high quality, secure, and also healthy. Okay, so this is what I want to show you also about the map here. Okay, this is about the what is the what the contributions of aquatic animal food to animal protein supplies. So briefly, I want to show you that the yellow dot, this is more than 20% contributions of aquatic animal food to animal protein supply in the world. Okay, so in any industry, they have a problem. So what is the problem? What are the problems in aquaculture? Okay, for me, this is a main problem in aquaculture. However, when you look at the statistics for today, the analysis shows that the feed is the major problem, particularly in the term of price. Because now is a raw materials like soybean, one of the ingredients that they formulate the feed is very expensive. Okay, but if you focus on the productions, so the reduction in production due to disease is a significant issue because some disease can lead to total loss. For example, last time is on 2019 to 2010, if I'm not mistaken. So we have problem with we start a problem with EMS in shrimp, so which is many industry got a problem and got a total loss too. Okay. However, given the current global condition, climate change is a major threat to aquaculture today and leading to emerging diseases. And then climate change is considered a threat to global food production in terms of both quality and also the quantity. And also the disease management and strategy is very, very important now, okay, on preventing and the development of infections that more important than treating disease stop. So the figure here, I want to show you the flow that when the disease occurred, okay, it will cause a high mortality, high morbidity, poor growth rate, and also low quality of flesh. For example, the low quality of flesh For example, the shrimp infected with fungus, for example. So it will cause the black spot disease and or black gill disease. So the appearance also is not good. Okay, poor appearance, and the quality of flesh also is not good. It's look like it's a soft. Okay, it's soft, and the taste also is not nice. Okay, when all this happened, it will be reduce the trade and reduce profit margin, which is led to cause the economic loss. So this is very important. You see that the flow here. So for me, is this is very important. And here is the World Fisheries and Aquaculture Productions. This is a brief, I got it from the FAO 2024 report. So the global fisheries and aquaculture production is about 223.2 million tons in 2022. And what I want to show here is a global aquaculture productions is about 130 uh, 0.9 million tons, and the capture fishery is 92.3 million tons. And the aquatic animal production by regions, when you see here, this is Asia, is about 70%, and compared to the another countries like Europe, 9%, Latin America and Caribbean is 9%, Africa 7%, and so on. And the main producer of aquatic animals by country, you can see here that the China is a lead, it's about 36%. India 8%, Indonesia 7%, Vietnam 5%, and Peru 3%. So of the total production of aquatic animals in 2022, it's about 62% was harvested in marine area, which is 69% from capture fisheries and 31% from the aquaculture. And if you see that inland waters is about 38%, which is 84% from aquaculture and 16% from the capture fisheries. And so I'm focused directly to the shrimp industry. So Asia leads the world in cultivated shrimp production, like China leads the world in shrimp production, followed by India and also Indonesia. Actually, last time, Thailand has historically been one of the world top shrimp producer, but in recent year, it has surpassed by China, India, and Indonesia in terms of total production volume. However, Thailand is still is top five shrimp producing countries in the world. So a major constraint limiting the shrimp production is a disease, as I mentioned before. Okay, it is estimated that approximately 60% of disease losses in shrimp aquaculture has been caused by viral pathogen, 20% by bacterial pathogen, and 20% by variety of other pathogens like parasite, fungus, and so on. And about 20 non-viral pathogen in shrimp. Okay. And then, however, sick viruses are responsible for the main considerable economic losses, which is white spot syndrome virus. This is still occur right now. And TSV, Taurus syndrome virus, HPV, hepatopancreatic parvovirus, IHHNV, infectious hypoderma and hematopoietic necrosis virus, MBV, monoderm baculovirus, and the YHV is yellow head virus. Okay. Before we go further to discuss about this topic, about this case study, so I want to show you that the shrimp most common farm it is Lithopenes waname, which is a white shrimp, and Pinus monodon, this is a tiger shrimp. So, in diagnostic, actually, we must know that what is an important organ, okay, for the shrimp, and then we can focus. And then what is the important organs that always, all the disease, we can detect it from that organs. Okay, that is hepatopancreas, that is the main organs, and also lymphoid organs. And lymphoid organs is only from the peanut shrimp that we can see. Okay, so before we go further, also we must know that the normal structure of the hepatopancreas, which is most of the disease, you can see it affect the hepatopancreas, which is a hepatopancreatic tissue showing that two zones here. Okay, the first is near the apical ends is E cells. Okay, and then the second one is the zone near the basal end is B cell of F cells and R cells. This is a very important cells and they have their own functions in the digestive. And this is a real one. So you can see that the E cells, okay, dominate with the E cells at the apical. And this is the tubules, okay, tubules for the hepatopancreas. So this is F cell. B cell and R cells. Okay, so the morphology of the cells also different. You can see here that like B cell is a big cell. Sometimes you can see that it's like vacuoles or something like that. Okay. And okay, so we go through general procedure for diagnosis for all the diseases from the shrimp. This is a general procedure. So it's maybe you have another investigation that you want to do. First is the history. So the history, meaning that you maybe ask the owner that where you get the PL post larvae. Okay. And then, or you, where you get the broodstock, for example. So, and then what's the history of the disease occurred from this farm, for example. That, and then after that, you go to the cross observation where you get the sample. So you cross the observations. Maybe in the farm, you can see that the behavior of the shrimp. Maybe they react or not with the feed given. Okay, and then is it lateral swimming or is it just keep quiet and then weak? Okay, and then you can see also the mortalities. You can ask the owners like how many percent of the mortalities occurred in the first day, second day, and third day, or in the first day is already 100%, something like that. And the feeding, so what feed, and then also response of the shrimp to feed and so on. And then for the external observations also you must you when you get your samples, okay, you observe the color. So sometimes the color can tell you that what possible disease. Okay, for example, the color change to the red, it could be the red disease syndrome, maybe, okay, or it could be the vibrio infections. Okay, and then soft shells. It could be the rostrum. Sometimes the rostrum also. The rostrum is a hard, okay, hard. But with the disease, okay, the rostrum sometimes is soft, okay, and sometimes the body also soft. Okay, the body, the shells is soft, and then shell damage. For example, cell damage is one case that I handled before that very ugly shrimps look like because it's a black color erosions on the shell. Okay, that's it cause a shell damage because of the fungus infection. And then also the black spot. Okay, then also the black spot. And then the environmental parameters. This is also important because sometimes the salinity changes also can harm to your shrimp. Okay, the pH, temperature, human activities also, and also the predators. All of these factors can cause stress to the shrimp. When the shrimp stress, so many diseases can occur because the immune system was down. Okay, and the pattern of mortality. Okay, is it localized or widespread? So this is you must think about. And feed and type of feed and feeding activities, how's the feeding activities at that farm. Okay, and also any chemical treatment used before or at that time any chemical treatment used. Okay, so this is very very important that you must take note. And okay, and then lastly the record keeping, that you want to see that disease management and form management and also the treatment that given before. okay, to the farm. Okay, this is a case study that I've choose for today is a hepatopancreatic parvovirus infected penis monodon. Okay, why I'm choose this is quite interesting for me because I trace from the grow out pond, I trace from the hatchery, and I also trace that from the broodstock. Okay, so in this case, we go to the hatchery phase first, and then for the history, so this is a Pinus monodon, which is a post-larvae 4 to 15 from one farm. And the conditions of PS is good, okay, and mortality is not significant, okay, then the clinical sign is no clinical sign. Okay, and then we took samples, okay, 10 PLs. Sample were collected from each stages of PL, PL4 and PL15 at this time. And then fixed in Davidson solution for 24 hours. Why I colored this fixative solutions and also the time because this is suitable for the PLs. Okay, this is based on my experience. I want to share with you today. And after 24 hours, then we place to 70% alcohol. Okay, and then we follow the standard method for the tissue process. Okay, and then after we sectioned, okay, and then we stained using the hematoxylin and eosin. This is a common staining that used for the to stain the histology tissues. Okay, and this is a one PL PS2 small. Okay, you can put the whole shrimp on one slide. So this is the result. Okay, so this is a total area of the hepatopancreas. Okay, this is the whole body of shrimp on the glass slide. So when I focus, zoom in for the PL4, this is a PL4, the early, we are detect the early in development as a small and eosinophilic bodies here. You can see that's a red arrow. And then this one is I stain with the hematoxylin and eosin. So you can see that the difference of the eosinophilic bodies here. And when you zoom in again, so PL4, this is a basophilic inclusions bodies in the hypertrophic nuclei in the hepatopancreas tubule epithelial cell. This is a tubule epithelial cells, okay? This is a tubule epithelial cell. So you can see that the inclusions body here at the pointed with the red arrow. Okay, this is a hepatopancreatic parvovirus infections. So these conditions. And the conclusion for this hatchery phase, the shrimp from the hatchery phase, PL4 and 15. So the presence of a single large basophilic inclusion bodies in hypertrophic nuclei in the hepatopancreas tubule epithelial cells, I can confirm the HPV. Okay, this is from the histology part. And then for the molecular part is already confirmed about it. That's what I want to discuss and shows that how the histology look like, histopathologies look like. Okay. High numbers of inclusion body was detected in PL4 compared to PL15, and PL15 shows that severe hepatopancreatic necrosis, but no necrosis was observed in PL4. And shrimp infected with HPV usually show non-specific growth signs of disease because we didn't see any signs. Okay, at the early juvenile stage, shrimp showed stunted growth and began to die. So there is the owners to complain. Okay, because it's stunted growth. Okay, and then okay, we go to the grow out phase. Okay, the grow out phase for HPV, this one, uh, age is juvenile, it's about PL30, and condition is poor. Okay, mortality, first day is 30%, second day 30%, and the third day is 100%. So after 8 weeks culture in ponds. Okay, the clinical signs vary in size of juvenile shrimp. Okay, weak with a rotten tail and also black spot on the carapace and the body. And we observe it Okay, we examine it, and then what I highlight again here that is 10 juvenile sample was collected from infected pond, fixed with the Davidson solution for 48 hours, because you know that this age the shrimp is a bit bigger. Okay, it's not too big, but it's a bit bigger and then harder. Okay, so you must use fix in the Davidson with a uh, about 48 hours, not more than 48 hours, because if you extend to 72 hours, you will be damaged the tissues. And if you let's say you said that this is a small shrimp, so you want to fix it in the Davidson's about 24 hours, but sometimes it's not fixed well. So you must think about it. And then sometimes it depends on the size of your shrimp also. This is based on the experience. What I did is I put in the Davidson solution for 48 hours for the juvenile is a good result. Okay, and then after that I put in 70% alcohol and follow the process procedure for this histology process, and then stain by using the HNE stain. This is a common stain. So we can see that this is a total area of the hepatopancreas of this juvenile, and then we zoom. Okay, we can see that the sign of necrosis and granuloma here and atrophy of the hepatopancreas. Okay, so when we zoom in, okay, then you can see the very clear granulomas here and also the necrosis of tubule epithelial cells. This is the tubules, so you can see that the necrosis occurred here. So you cannot see that very nice tubules with the cells that I mentioned before, the B cell, R cells, and then E cells. You cannot detect it because it's already necrosis like this. And this is what I want to show at the different field of the problem shrimp from the grow out phase is atrophy of the tubular epithelial cells. So this lesion is related to starvation, anorexia, and also poor because of the poor growth rate. So you can see that the poor development of the hepatopancreas, especially the tubular epithelial cells here. So you can see that the B cell is not active. The B cell, which is I focusing a point with the red arrow. So not active to produce and secrete digestive enzymes. So this is shows that the starvations and or anorexia. Okay.
I'm sorry, Professor.
Yeah.
You have 5 minutes left for your presentation.
It's okay.
Yeah, okay.
Okay. Yeah,
Okay, thank
enough,
you,
I think.
Professor.
Okay. So you can see that here is a sloughing of epithelial cells and hemocyte infiltration. So here that you can see the hemocyte infiltration is a lot, but I'm not focused to zoom in. Okay. And then is it the necrosis because of the virus or bacterial toxin? Because in this case also, because in this case, when the viral infection, HPV especially, so it's also that mixed with the bacteria like Vibrio species. Okay, the conclusion for the grow out phase is the presence of a single large basophilic inclusion body in hypertrophic nuclei in the hepatopancreas tubules epithelial cell. This is confirmed the HPV. And a high number of inclusion bodies was detected in the hepatopancreas. Okay, the shrimp showed severe hepatopancreatic necrosis. At PL30, the shrimp started to die. So the shrimp infected with HPV usually show non-specific growth sign of disease. At the earliest juvenile stage, the shrimp showed stunted growth and began to die. So this is a different with the PL4 and 15 to 15 just now that I'm present, because at this stage is we detect that's a lot of necrosis, hepatopancreatic necrosis occurred. So this is we move to the broodstock because this is happen where it's come from. So I trace where they get the seed. So we go to the broodstocks, and then we see that the broodstock, everything is good for the broodstock, except the clinical sign is rotten tail and shell and head. At tail, shell, and the head area, but this is based on the management. Sometimes bacterial infection, sometimes because of the injuries. Okay, so this is a very important. I highlight here because the broadstock is so big. Okay, so we use the technique of injections of Davidson solutions. And then after turn it to red color, so we are cut into two sections because we want the Davidson solution can fix the hepatopancreas well after that, and then also the muscles. And then we are placed into 70% alcohol, and then follow the another standard method for the uh, histology process, tissue process. And then this is the result. Very interesting. That is a male P monodon. So you can see that the section of hepatopancreas tubules, this is here, show that the presence of the basophilic inclusion bodies in the hypertrophic nuclei of hepatopancreas tubule epithelial cells. It's not many, I found the inclusion bodies, but it have that. And then the HPV inclusions often push nucleolus against the nuclear membrane. And then here you can see here that this look like crescent shape. It's always happen. Okay. And this is the characteristic of HPV infections. And then the hypertrophic nuclei and vacuolizations, you can see here, of the cells also observed from the samples. And for this one, the section, you can see that the tubule of pimonodon with intranuclear inclusions body also. Okay, this is stained by the with the HNE stain. And this is a section of hepatopancreas tubules of pimonodon with hypertrophic nuclei and vacuolizations of the cells. This is when I zoom it, as you can see, that is the process of infections. Okay, so the for conclusions, okay, the male only a few cells contain inclusion bodies, and the nuclei were hypertrophic, and there was vacuolizations of the cells. The gills, intestine, and muscle were normal. And female, the section hepatopancreas shows that no inclusion bodies, but it shows hypertrophic nuclei and vacuolization of the cell, maybe the process, okay? And the cause of hypertrophic nuclei was unknown, okay? The gill, intestine, and muscle were normal. And I can conclude that the histopathological observation showed that the lesions in male shrimp were typical of HPV infections, and hypertrophic nuclei of hepatopancreatic tubular epithelial cells observed in both cases could be due to viral activity. And for general conclusions, that I can conclude that HPV infected incultured shrimp cause a chronic mortalities during early larval stage or post larval stage and juvenile, and infected shrimp showed non-specific gross sign of disease. Early juvenile stage showed that stunted growth and mortality. This is always the stunted growth. This is very, very clear. You can see. And then histopathology is one of the methods that useful for detecting the viral infection, especially in broodstock, which may act as a carrier because they use the PCR for the broodstock. That is a negative. Okay, this is only positive for HPV when they check for the PL. Okay, so thank you. I thanks also to my colleague who are involved in this project. Okay.
All right. Thank you very much, Professor Marina, for that insightful presentation about viral incidents in shrimp culture, especially about hepatopancreatic parvovirus. I'm sure our audiences has found it very informative. Thank you, Professor Marina.
Okay, you are welcome.
Okay, thank you, Professor. Now let's move to our next esteemed speaker, Mr. Abdul Aziz Jaziri, SPIMSC, and allow me to briefly introduce Mr. Abdul Aziz curriculum vitae. Okay, Mr. Abdul Aziz Jaziri, SPIMSC, is a lecturer in Faculty of Fisheries and Marine Science, Universitas Brawijaya, and he is currently completing his doctoral degree at the University of Malaysia, Sabah, in the Faculty of Food Science and Nutrition, and then master degree in Department of Biotechnology, Graduate School of Life Sciences, Ritsumeikan University, Japan, and also bachelor degree in Faculty of Fisheries and Marine Science, Universitas Brawijaya. Next. Okay, with his career spanning roles in project management,